So moving on to the first cocci, there are 5 cocci of pathogenic importance. One is staphylococci. The second one is streptococci. The third one is pneumococci. The fourth one is meningococci. And fifth one is gonococci. Okay. Why they are called staphylococci? Staphylo means grapes. And cocci, they are all spherical. Bunch of grapes and they are spherical in shape. They are bunch of grapes, they are spherical in shape. Streptococci is chain. Staphylococci, they are bunch of grapes arranged in clusters. Strepto is chain, serpent, chains, serpent. Pneumococci is plano-convex and 2 cocci enclosed in a single capsule. Meningococci, these 3 are gram-positive. And these 3 are diplococci. Okay. Meningococci and meningococci and gonococci and pneumococci are diplococci. These 3 are gram-positive and these 2 are gram-negative. Okay. Now, meningococci is plano-convex, half moon, and gonococci is always seen intracellular in the inside the kidney shape. Inside polymorphonuclear neutrophils, they become pus cells. So I will repeat once again: staphylococci, gram-positive cocci arranged in clusters; streptococci, chains; gram-positive cocci arranged in chains; pneumococci, plano-convex, plane surface facing each other; meningococci, plano. So this is lancet shaped, the broader end facing each other. Meningococci is plano-convex, plane surface facing each other. Gonococci kidney shaped, predominantly they are seen inside. First 3 cocci are gram-positive, next 2 cocci are gram-negative. So that is a basic understanding of these 5 pathogenic cocci which cause infection in the man. Now coming to the staphylococci. Staphylococci is staph. It's a gram-positive cocci. Gram-positive cocci arranged in clusters. It's non-motile, non-sporing, non-motile, non-acid fast. They look like bunch of grapes because they divide in all planes. and daughter cells fail to separate out from the mother cell. Daughter cells fail to separate out from the mother cells. They looks like a grapes. Okay. Now moving on to the moving on to the important characteristics of staphylococci. They produce pigment, non-diffusible pigment. The colonies are colored. So chemically they are made up of beta carotene, non-diffusible pigment, non-diffusible pigment, and they depending on the pigment production that can be enhanced by milk agar and milk salt agar is a it is a salt tolerant organism. Milk salt agar is used for growing. It is a selective medium for growing staphylococci. Selective medium for growing selective. Now, depending on the pigment production, staphylococci are classified as aureus, golden colored, golden yellow. Remember, king with gold crown. Golden yellow color, albus. Albus, remember egg white, and citrus. Citrus, lemon. Well, they look orange. Citrus fruit, orange. They are classified into aureus, albus, and citrus. So there are other organisms which also cause infection. That is epidermicus, and saprophyticus. Epidermicus and saprophyticus. Epidermicus and saprophyticus. Okay. Now moving on to they are non-fastidious organisms. They can grow on nutrient agar, blood agar, milk salt agar, they show they are salt tolerant. On blood agar should beta hemolysis. They say it is oil paint appearance on nutrient agar. So you should remember staphylococci for two C's. This is staph. What are these two C's? One is catlase, another is coagulase. Catlase and coagulase. Catlase is enzyme produced by staphylococci which converts hydrogen peroxide into water and oxygen. So you take a saline suspension of staphylococci, pour H2O2, there will be bubbling. This test is used. This test is used to differentiate between staphylococci and streptococci. Staphylococci is catlase positive. Streptococci is catlase negative. Next is coagulase. What is coagulase? Coagulase enzyme produced by staphylococci, which converts fibrinogen into fibrin. So it protects against phagocytosis. Protects against phagocytosis by forming fibrin barrier around the organism. It protects the staphylococci by producing fibrin barrier around the organism. So there are two types of coagulase. One is free coagulase, another is bound coagulase. Free coagulase and bound coagulase is a part of cell wall. Bound coagulase is a part of cell wall. Bound coagulase is a part of cell wall. Free coagulase, it is diffused into the medium. Okay. Now, how do you test free coagulase and bound coagulase? The bound coagulase is tested by slide coagulation test. Bound coagulase is tested by slide coagulation technique. We take a saline suspension of the organism and add plasma. There will be clumping. Of course, you have to take the control also. That is the slide coagulase test, which detects the coagulase bound to the cell wall. The next is tube coagulase. What is tube coagulase? Tube coagulase tests the free coagulase. We take saline suspension of the organism, add 1 in 10 diluted plasma, incubate at 37 degrees for 4 hours. Incubate at 37 degrees for 4 hours. So after the 4 hours, look for the thick coagulum at the bottom of the tube. So it needs the help of CRF, coagulase reactivating factor. It needs the help of coagulase reactivating factor. Now, moving on to the pathogenicity and clinical infections, laboratory diagnosis of staphylococci. Staphylococci is, we all carry staphylococci in the nasopharynx. %40 of hospital personnel and healthcare workers carry, and approximately 20% of normal people carry. And there is something very interesting. We all hospital people, doctors carry MRSA. We'll tell you about MRSA. That is methicillin adhakifanac resistant staph aureus. The root, okay. ಏನಿಲ್ಲ ಡಿಸ್ಟರ್ಬ್ ಏನಿಲ್ಲ ಮಾಡಲಿ ಮಾಡ್ತಾ ಇದ್ದಾರೆ ಅದು ಆಫ್ பண்ணிட்டு ಅದೆಲ್ಲ ಡಿಸ್ಟ್ರಾಕ್ಟ್ ಅಂತ ಕ್ಲೀನ್ பண்ணி ಆಮೇಲೆ ಆಪರೇಟ್ ಮಾಡ್ತಾರೆ ಅದು ಔಟ್ಸೈಡ್ ತೆಗೆದ್ಬಿಟ್ರೆ ಏನು ಪ್ರಾಬ್ಲಮ್ ಆಗುತ್ತೆ ಏನಾಗಲ್ಲ ಅವ್ರು ನಮ್ಮ ಕಾಲೇಜ್ನವರೇ ಅಲ್ಲ ಏನಿದೆ ಅದರಲ್ಲಿ ನಾಗರಾಜಿಗೆ ಫೋನ್ ಮಾಡಿ ಹಾಂ ಫೋನ್ ಮಾಡಿ ಕೇಳಿ ಫ್ಯಾನ್ ಹಾಕಿ ರೆಕಾರ್ಡಿಂಗ್ ಆಮೇಲೆ ಆನ್ಲೈನ್ ಯೂಟ್ಯೂಬ್ನಲ್ಲಿ ಬಿಡೋದು ಆಮೇಲೆ ಯಾ ನಾವು ಸೊ etiopathogenesis is when there is a breach. Our patient is diabetes mellitus, immunocompromisation, viral pneumonias, surgical site infection, burns, cuts. All this, when there is a breach in the skin, the staphylococci is going to invade and cause infection. So remember, staphylococcal infection. So it causes variety of lesions. Remember, first when you are sitting in the viva, they will ask what are the infections caused by. Suddenly, student becomes blank. They don't know how to answer. Remember at least ABC. Abscess, boil, abscess, boil, and carbuncle. Abscess, boil, and carbuncle. Please remember this: abscess, boil, and carbuncle. So it infects almost all parts of the body, human body, whenever it gets an opportunity, because it's quite abundant in the nasopharynx, and it is most commonly transmitted by healthcare workers and by contact and aerosol if it's causing pneumonia and abscess. So now first we start with the skin lesions. Skin lesion, it causes boil, carbuncle, impetigo, abscess. So variety of skin lesions: nail bed, infection of the nail bed, folliculitis, stye. So variety of skin lesion it can cause. Now coming to the respiratory, it can cause any infection, tonsillitis, pharyngitis, laryngitis, tracheitis, tracheobronchitis, bronchitis, and pneumonia. Cardiovascular, it can cause carditis and it can cause abscess and it can cause acute carditis. CNS, it can cause brain abscess. UTI, the most commonest organism is cons, coagulase negative staphylococci, along with other organisms. It can cause any infection of the urinary tract infection. So, very important organism. It causes pus. What is pus? It's a end result of battle between bacteria and the phagocytes. So it produces. They are called as staph and strep pyogenes. So it causes variety of clinical infection. We are only bothered about this surgical site infection and infection of the burns. That's very important. People die of uh, infection after burn itself. Actually, that is a third or fourth day they gets infected, burn it often, and gives rise to infection. So, what are the various virulence factors? It produces hemolysis, as you said, beta hemolysis, leukocidins, Mantin Valentine factor. It produces enterotoxin, and various other virulent factors. Important. It is also exfoliative toxin. So various other toxins are produced. It produces beta hemolysis. On blood agar. It produces beta hemolysis on the blood agar. Very important. You should know. Okay, that's very important. So now you know man get infected by contact or aerosol when there is a breach and there will be pus, abscess, boil, carbuncle, furuncle, surgical site infection, post-operative wound infection, abscess, pus, and all those things. So lab diagnosis. No, very important is non-suppurative infection by staph. Non-suppurative infection with staph. Please remember three, very important. One is food poisoning. Second is toxic shock syndrome. SSS. Staphylococcal superficial skin scalded syndrome. This occurs due to the toxin. Enterotoxin and exfoliative toxin. Enterotoxin and exfoliative toxin. So, very important is lab diagnosis. Food poisoning is described as meat, poultry product, incubation period is 6 hours, self-limiting oral rehydration, and vomiting, diarrhea. vomiting and diarrhea. Toxic shock syndrome, very severe due to infected tampons, which ladies use during menstrual cycle, and it gives rise to super antigen and complement action, and sometimes it leads to death. And SSS looks like the dry burns seen usually in children. So these are all non-secretory infection caused by toxins released by staphylococci. Please remember uh, FTS, FTS, mnemonic. F for food poisoning, T for toxic shock syndrome, SSS. Remember like that. The laboratory diagnosis, whatever the sample you receive, usually it's a pus swab. Do gram stain. Gram stain, you see plenty of pus cells and gram positive cocci arranged in cluster. Okay. Now, then you do catlase and coagulase to confirm this staph RS. Okay. Now, how do you find out MRSA in the lab? The cefoxitin disc is used. The other antibiotics are put, and next day look for the antibiotic zone of inhibition and measure with Capriber. If it is cefoxitin resistant, the surrogate marker for methicillin. Why do you ask me why methicillin was not used? Because methicillin shelf life of the disc is very short. Cefoxitin, the shelf life is very short. We use so MRSA drug of choice is vancomycin, linezolid, oral. Now, what are the modes of drug resistance of staphylococci? What are the modes of drug resistance of staphylococci? Number one, it produces beta lactamase and it destroys the beta lactam ring. So, beta lactamase and alteration in the penicillin binding sites, penicillin binding proteins, PBP. And third one is penicillin binding protein. Third one is development. It will not allow the drug inside. And fourth one is development of tolerance. Penicillin has become totally useless for the treatment of staphylococcal infection. Always we use BLIs, beta lactam inhibitors. There are three. Remember with TCS software company, tazobactam, calonic acid, salbactam. Tazobactam, calonic acid. So you must have heard of a drug called amoxiclav. Amoxiclav. So please remember this. is always taken for staphylococcal infection, the drug of choice for the treatment of staphylococcal infection. Okay, now moving on to the fast typing. What is fast typing? It's important for epidemiology. Find out the source of infection. The barge is a tadpole-like structure which is present on the parasitic life on the on bacteria. It produces lytic cycle, which is seen in staph, and lysogenic in C. diff. It gets integrated into the nucleic acid beta phage, which is responsible for toxicity, which is responsible for toxicity of staphylococcus. Okay, now what you do is, for example, remember hospital ward or post-operatively. So remember the hospital ward. Remember this is a ward. There are about 3 beds. Post-operatively they are operated. All of them are infected post-operatively with staph, surgical site infection. Whatever may be the operation, all of them are infected with staph. So we want to know who is the source for this patient getting infected, whether it's a surgeon. What is the whether it's a surgeon or OT technician or nurse or a person who is assistant. So we take swab from nasopharyngeal swab from all these people, and this staff we send it to Maulana Azad Medical College, New Delhi. Maulana Azad Medical College, New Delhi. So look for the fast typing. Usually, if they are found similar in surgeon and all these patients, then we'll ask surgeon to go on a leave for 6 to 8 weeks and treat his staff carriers. When he gets rid of staff carrier status, then he is allowed to get back to do surgery. So this is a importance of fast typing, so which is done in Maulana Azad Medical College, New Delhi. So whenever you get a staph infection, whole ward post-operative ward get infected, all the patients get infected, you have to do the fast typing source infection and ask the person to leave and get rid of the staph which is carrying and treating. Okay, that is about staph. Important thing, staph is it's a hospital bug. Carriage is 40% in healthcare workers, and it has become penicillin resistant, and there is MRSA, methicillin staphylococcus aureus, treated with vancomycin and linezolid, and so periodically you have to do MRSA carrier status in hospital care workers, and treat them with mupirocin nasal pack and chlorhexidine soap. so that they do not carry this MRSA, and whole chapter can be taken on MRSA carrier and all this. So important points to remember as far as your examination is concerned: pathogenicity, lab diagnosis. They can ask anything like food poisoning, toxic shock syndrome, or SSS. All these things can be asked as a short notes. And coagulase test. Again, one more thing important I forgot is mannitol motility medium. Staphylococcus is mannitol fermenter. It is non-motile. If you see you inoculate into the mannitol motility medium, it shows straight line, color get change. So mannitol fermenter, non-motile. With this, I leave you. Read and come back for the next class on streptococci. Thank you very much. God bless you. For offline classes, you can contact me 98451 33798. Dr. Suresh Bhogi. Professor, HOD, PS, Medical College, Kupang. 98451 33798. Thank you very much.